Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Excel International GCSE Biology Paper 1BR for May 2024. Let's begin with the first question. In question 1, the image shows some cells from onion epidermis. So these are the cells. We can see there is a cytoplasm, we have cell walls and the nucleus. Then they say some structures in these onion cells are also found in human red blood cells. Which row in the table is correct for human red blood cells? Human red blood cells have no cell wall, they have a cytoplasm and there is no nucleus, so the answer is C. Next, they say the onion cells in the image are all epidermis cells. What is the term for a collection of cells of the same type? That is a tissue. Then they say the images show samples of red onion epidermis cells before and after being placed into a 5% sucrose solution for 1 hour. So if the cells are placed in this solution, it means they're going to lose water because this is kind of more concentrated than the cytoplasm. And here we can see the cell membrane has detached from the cell wall as a result of water being lost out of the cell. So they say describe how to make 10 centimeters cubed of a 5% sucrose solution from a 10% sucrose solution and pure water. So in this case, we are using the 10% solution to make a 5% solution, and the volume is 10 centimeters cubed. So we can take 5 centimeters from this and dilute it with 5 centimeters cubed of distilled water. So I said use a syringe, add 5 centimeters cubed of pure water to 5 centimeters cubed of the 10% sucrose solution. That way you make double the volume, or 10 centimeters cubed of half the concentration. Next, they say explain the change in appearance of the onion cells shown in the images after being in the 5% solution or 5% sucrose solution for 1 hour. If they are in this solution for 1 hour, they will lose water by osmosis and they will become flaccid. So I said water leaves the cells by osmosis towards the sucrose solution from high water potential to low water potential, and the cells are going to become flaccid. In question 2, the diagram shows the structure of an insect-pollinated flower. We can see the flower here. These are the anthers, we see the stigma, and so on. They say which part of the flower produces pollen. Pollen is produced from Y, the anthers, so the answer should be C. And next they say which part of the flower becomes a fruit. The ovary becomes the fruit, so it should be V. And next they say the image shows a pollen grain produced by the plant, which is this one here. The diameter of the pollen grain in the image is 30 millimeters. The pollen grain has an actual diameter of 313 micrometer. Calculate the magnification of the pollen grain in the image. They want you to give your answer to the nearest whole number. So we can see this is the image, which is 30 millimeters, and here they've given us the conversion. So that should be 30,000 micrometer. So 30,000 micrometer divided by the actual, which is 313 micrometer. Again, the units have to be the same so that they can cancel out since magnification has no units. So I got 95.8466, but they want to the nearest whole number, and my answer was times 96. Then they say state how the structure of the pollen grain is adapted to ensure insect pollination. We saw here it has spikes, so I said it has spikes. The next they say gardeners often reproduce plants by taking cuttings. Explain why gardeners often reproduce plants by taking cuttings instead of growing plants from seeds. Because seeds come from gametes which are produced by meiosis, the seeds are going to have variation in DNA, but when we use cuttings, we create plants that have the same DNA or that are genetically identical, and therefore they're going to have the same characteristics. So I said using cuttings is a form of asexual reproduction. The offsprings are genetically identical to the parents, and the farmers can therefore obtain many plants with identical phenotype. And actually, these plants can be maintained within a shorter period of time. Next, they say the diagram shows a piece of apparatus used as a model for breathing. The rubber sheet can be pulled down and pushed up to represent breathing. We can see here the balloons representing the lungs, the glass jar, and so on. They say name the structure that the rubber sheet represents in a human. This represents the diaphragm. And next they say which row in the table gives the changes in volume and pressure inside the glass jar when demonstrating inhalation. So this should be when inhalation takes place. the volume is going to increase and then the pressure is going to decrease. So the answer should be C. Next, they say give 2 reasons why the apparatus does not give an accurate representation of human ventilation. If we go back here, we can see this is a glass, but in the chest cavity, we have rib cages as well as intercostal muscles that can allow for expansion, but the glass is rigid. So I said there are no rib cages and there are no intercostal muscles. In part B, they say the diagram shows the structure of an alveolus. We can see the capillaries and the inside of the alveolus, the thin distance allowing carbon dioxide out and oxygen in. And they say which of these is the order of the structures that air passes through from the mouth to the alveolus during inhalation. So from the mouth, it goes to the trachea, then the bronchus, and then bronchioles and the alveolus. So the answer should be D. Then they say the total surface area of the alveoli in the lungs is very large. A typical human lung has 480 million alveoli. Each alveolus has a surface area of that. Calculate the total surface area in millimeters cubed of the alveoli in one typical human lung. They want to give your answer in standard form. So this is easy, just multiply that by that. And I got about 1.92 times 10 power 6 in standard form. Then they say explain 2 ways that a single alveolus is adapted to maximize gas exchange. It's made up of 1 cell thick, or you can say it's 1 cell thick, providing a short distance for the gases to diffuse. It is moistened to allow gases to dissolve before they diffuse into the blood, and it contains many capillaries to provide a steep concentration gradient. Then they say describe a simple laboratory experiment to investigate the effect of exercise on the rate of carbon dioxide release from humans. In this case, we can get 2 participants that are of the same age, same gender, and then we can use lime water. You could make them breathe through lime water or hydrogen carbonate indicator, and then measure the time it takes for maybe the lime water to turn cloudy. So I said ask a person to breathe into a test tube containing lime water before and after exercise. So the exercise could be jogging for about 2 minutes. Then you measure the time taken for the lime water to turn cloudy in both cases, indicating carbon dioxide release. Then you repeat the experiment with several different people to ensure reliability and account for variation between individuals. While doing this experiment, people should be the same age, same level of fitness, and same gender. And the lime water should be the same volume and concentration. Question 4. A student uses this method to investigate the effects of temperature on the digestion of starch by the enzyme amylase. They place 5 centimeters cubed of starch solution into a test tube, and then place 1 centimeter cubed of amylase solution into a second test tube. They place both the test tubes into a water bath at 10 degrees Celsius for 10 minutes. Then they pour amylase solution into the starch solution and mix. So this placing both test tubes is to ensure that they are both at 10 degrees before they are mixed. Then they test a sample of the mixture for the presence of starch every 5 minutes until no more starch is present. They repeat at different temperatures, as we can see here. So they said the investigation is repeated 2 more times at the temperature, that is to find the mean. The first part says what is produced when starch is digested by amylase. Maltose is going to be produced. Then they say which of these parts of the human alimentary canal produce amylase. The colon does not, the pancreas does, and the salivary glands do. These produce the salivary amylase and that the pancreatic amylase. So the answer is 2 and 3. Then they say state why the test tubes are placed in a water bath for 10 minutes before mixing the contents. This is to ensure that the temperature is 10 degrees before amylase and starch are mixed. Then they say describe how the student could test a sample of the mixture for starch. In this case, you use iodine solution and observe the color change. If it turns blue-black, then starch is present. Next, they say the table shows the results of the student's investigation. So we see the temperature variations and the time taken for the starch to be digested for the different experiments. So they're asking us to find this mean value here. So they say calculate the mean time taken for all starch to be digested at 30 degrees Celsius and give your answer to 2 significant figures. So I added that plus that and divided by 3, giving me 23. to two significant figures. The next they say explain the effect of increasing the temperature from 10 to 40 on the time taken for all starch to be digested. If we go here, we can see the mean time decreases all the way to 40, but here we see starch wasn't digested, so at 50, there is going to be the denaturation of the enzymes. So from here to 40, the higher the temperature, the higher the rate of reaction because there is more kinetic energy. So I said increasing temperature increases kinetic energy, so the enzymes and substrates collide with more energy, leading to more successful collisions per time. And the optimum temperature is reached when the rate is the highest. Then they say explain why the starch was not digested when the temperature was 50 degrees. Like we already saw, at 50 degrees, the temperature is beyond the optimum, and the enzymes are denatured, so their active sites and the substrate are not complementary. Next, they say explain how the student could modify the investigation to give a more accurate measure of the temperature at which the amylase activity is fastest. If we go back, we can see the temperature decreased, but we did not know if the minimum was around here. So what we can do is repeat the experiment between 30 to 50 degrees using smaller intervals to see where we have the maximum rate or fastest rate of reaction. Next, they say the diagram shows part of a food web for the Arctic Ocean. So that is the food web, and down they say phytoplankton are prototists that photosynthesize. Which of the following features do phytoplanktons share with plants? So cell wall of chitin, no, that's wrong. Chloroplast, yes, and nucleus is okay, so 2 and 3 only. Then they send them to organisms in the food web that can feed as starch-free consumers. I use different colors to show the different pathways. This is producer, primary consumer, secondary, and tertiary, so the gray well. And then next, producer, primary consumer, secondary consumer, and then tertiary. So this one here is a tertiary consumer. And then we went to producer, primary, secondary, tertiary, also the seal. And then producer, primary, secondary, tertiary, so the polar bear as well. So the answers are polar bear, predatory fish, the seal, as well as the gray whale. Next, they say draw the food chain with the most trophic levels in this food web. And the one in blue, which is 1, 2, 3, 4, 5, and 6. So it had 6 trophic levels, as you can see here. Then in part B, they say scientists measure the changes in the biomasses of phytoplankton and zooplankton in 1 year. During a month in spring, the scientists found that the biomass of phytoplankton in the water increased from 1.2 to 12.6 milligram per decimeters cubed. Calculate the percentage increase of this phytoplankton in this month. So it changed from that to that, meaning the increase is going to be that minus that, giving us 11.4. So the percentage increase should be 11.4 divided by 1.2 times 100, giving us 950%. And that's the answer. The next they say during the year, the scientists also measure the change in the number of hours of light each day and the change in nitrate concentration in the water near the surface of the ocean. The graph shows the results, which we see here. On the vertical axis, we have relative amount of factor, and on the horizontal axis, we have the different seasons. Beginning with nitrate concentration, we can see it's higher in winter and early spring, and then it goes down as, of course, the others like the phytoplankton increase. Of course, they're using the nitrates. And we also see the zooplanktons begin to increase after the phytoplanktons have increased. That is because zooplanktons are eating the phytoplanktons. As the nitrate concentration goes down, it's being used. And the number of hours of light per day are fewer in winter, and they peak around spring to summer. So that's why we see a lot of growth of the phytoplanktons, because there is more light, more photosynthesis, and therefore increase in the biomass. So here they say discuss the reasons for the changes in the biomass of phytoplanktons and zooplanktons during the year. I've already explained this. The biomass of phytoplankton is low in winter but increases sharply in spring due to the increase in light intensity and longer daylight hours, which promote photosynthesis. This is supported by the graph where the hours of light per day rise significantly in spring. Additionally, nitrate concentration is high at the start of spring, providing essential nutrients for making amino acid, protein, DNA, chlorophyll, and so on. As phytoplankton biomass peaks, a zooplankton biomass starts to increase shortly after but there is a lag, and this lag is because zooplanktons feed on phytoplanktons. So their population increases as more food becomes available. The zooplankton biomass peaks in early summer after the phytoplanktons peak. Later in summer, phytoplankton biomass decreases due to both depletion of nitrates and increased grazing by zooplanktons. The zooplankton biomass also declines as their food source diminishes. We can see in autumn both biomasses drop to low levels due to shorter days, less light, and limited nutrient availability. We see as the nitrates are low already. In question 6, they say a reflex is a rapid response to a stimulus. The withdrawal of a hand when a finger touches a hard object is an example of a reflex. Describe the pathway of a nerve impulse in the reflex act involved in this response. So a stimulus arrives, the receptor is going to convert that to an electrical impulse, which is sent through the sensory neuron. The sensory neuron synapses with a relay neuron, and then the relay neuron synapses with a motor neuron. The motor neuron takes the impulse towards the muscle where they synapse at the neuromuscular junction. Between neurons, neurotransmitters are released to initiate the impulse in the next neuron. In part B, they say the photograph shows an African land snail. When an African land snail is touched on its eye stalk, the snail goes back into the shell. The snail then remains in its shell for a period of time. A student investigates the effect that repeated touching of the eye stalk has on the time the snail remains in its shell. So this is the student's method. They gently touch the eye stalk of the snail with a cotton wool bud so that the snail goes back into its shell. Measure the time the snail remains in its shell. After the snail has come out of its shell, they touch the eye stalk again so that the snail goes back into its shell. Then they measure the time the snail remains in its shell. They repeat the stimulus for 6 more times. Of course, the time is going to decrease with every touch because the snail is getting habituated. So they say the student's results are shown here. You can see as the number of touches increase, the time is going to decrease. Then they say plot the graph to show the effect of stimulus number on the time the snail remains in its shell. Join your points with ruled lines or ruled straight lines. So this is the independent variable which should be on the x-axis, and that is the dependent variable or the result to be on the y-axis. So I had time in seconds, as we can see there, and then here we have the number of touches or the stimulus number. So put the labels, put the units, put the axis in the correct position, get appropriate scales, and then points should be positioned appropriately, and just draw straight lines through the points in order to get full marks. Next, they say describe the effect of increasing stimulus number on the time the snail remains in its shell. As the number of stimuli increase, the time taken to remain in the shell decreases, and the greatest decrease is after 5 touches. Next, they say suggest a benefit to the snail of the difference in response to the repeated stimulus. It can be able to distinguish between dangerous or threatening stimuli and non-threatening stimuli so that it saves energy. Instead of returning into its shell, it can stay and feed. Also, if it's threatening, then it can learn to run away or to escape. So I said it allows the snail to escape from danger, and it allows the snail to distinguish between threatening stimuli and safe ones. So it can save energy by not returning into the shell if the stimulus is not threatening, and then it can continue to feed. In question 7, coronary heart disease, CHD, is caused by damage to the walls of the coronary artery. Explain 2 differences between the structure of the wall of the artery and the walls of the vein. So here we have to state the differences and explain them. The first part, I say the walls of the artery are much thicker or is much thicker than that of the veins because arteries have to withstand the high pressure of the blood being pumped from the heart, while the veins carry blood at much lower pressure. Also, the arteries have more smooth muscle and elastic tissue in their walls compared to veins. This allows them to stretch and recoil to maintain blood pressure. The veins have less elastic tissue as well as smooth muscle because the pressure is lower, and they rely on valves and surrounding muscles to prevent the backflow of blood. Next, they say the diagram shows a cross-section of a coronary artery from a healthy person and from a person with CHD. From a person with CHD, we see there is blockage in the artery due to the fatty deposits. So they say the heart needs to respond to extra demands during exercise. Explain why during exercise the heart of a person with CHD would not respond as efficiently as the heart of a healthy person. This person has blockage, so there is less oxygen and nutrients sent to the heart muscle, and so the heart muscle is not going to contract as desired during exercise. So I say in coronary heart disease, the coronary arteries are narrowed. During exercise, the heart needs more oxygen and glucose to meet increased energy demands. However, in CHD, the restricted blood flow limits oxygen supply. As a result, there is less aerobic respiration and less ATP, so the heart muscle cannot contract as forcibly or as quickly, so the cardiac output is reduced and the person may experience chest pain or fatigue. Next, they say genetics and smoking are two risk factors of CHD. The alleles of some genes increase the risk of developing CHD in people, so meaning there is a genetic component. These are known as risk factor alleles. Scientists compare the relative risk of developing CHD in people with different numbers of these risk factors. The scientists also compare the risk of developing CHD for non-smokers and smokers. Remember, the question is focusing on genetics and smoking. They say the graph shows the scientists' results. So here we see for non-smokers and for smokers. And for all risk factor alleles, we can see there is a higher risk of CHD among smokers than non-smokers, because we can see here higher, and higher. And for all, as the number of different risk factor alleles increase, the risk of CHD increases. Down here they say give one risk factor for CHD other than genetics and smoking. It could be gender, level of exercise, high cholesterol, high salt intake, obesity, diabetes, and so on. Then they say comment on the relationship between the number of risk factor alleles and smoking on the risk of developing CHD. They say use data from the graph to help you answer. You need to know that chemicals from the smoke can irritate the blood vessels, causing the blockages that could result from that, and also carbon monoxide and the other particles are connected or have a risk component to CHD. So I say smoking is a risk factor of coronary heart disease. Carbon monoxide and smoke particles cause CHD. As the number of risk factor alleles increase, the relative risk of developing CHD increases for both smokers and non-smokers. We saw this from the graph. For non-smokers, the relative risk increased from about 1 to 2. For smokers, the relative risk is higher at all levels. With fewer alleles, it's around 1.9 and high alleles about 2.8. This suggests that both genetic factors and smoking independently increase CHD risk. So smoking leads to blockage of the coronary arteries, so it further elevates the risk and showing a combined effect with genetics. The highest risk occurs in people who smoke and have high number of risk alleles. But there is no indication of sample size, gender, age, frequency of smoking, or the number of times or the number of packets of cigarettes these people smoke, so this could affect the conclusions that could be made on these results. In question 8, they say lactose is the sugar found in milk. The lactose needs to be digested in humans by an enzyme called lactase so the products can be absorbed. Or human babies produce lactase in their intestines. People who are lactose intolerant stop producing lactase and are unable to digest lactose sugar as adults. Lactose intolerance is a genetic condition caused by a recessive allele D. The ability to digest lactose is caused by a dominant allele, capital D. So the diagram shows a family tree. Remember, this is a male with lactose intolerance and this is a female without lactose intolerance. So if it's caused by a recessive allele, this should be small d, homozygous recessive, but we cannot yet tell the genotype of the mother. However, when we look at the offsprings, if these two parents had a homozygous recessive child, then it means the mother is heterozygous. So capital D small d, and this is small d. Therefore, this person here must be heterozygous as well because the father donates a small d. And if they're not lactose intolerant, then it means they are heterozygous. So down here they say use a genetic diagram to determine the probability of individual 4 and 5 having a child with lactose intolerance. We already know the genotype of these two individuals, so we can use that to create a genetic diagram. So capital D small d and small d gives us this is heterozygous, homozygous recessive, heterozygous, homozygous recessive, and the probability is going to be 50%. Then next, people with lactose intolerance often get diarrhea, which is a production of feces with high water content if they drink milk. Suggests why people who cannot digest lactose get diarrhea if they drink milk. This could be because the lactose is not digested, and so it's not absorbed, so it ends up in the colon or the large intestine, where there is a higher concentration, and so it draws water from the tissue surrounding, creating high volumes of water or high water content within the feces. So I said people who are lactose intolerant do not produce the enzyme lactase. So lactose is not broken down and passes into the large intestine, where it draws in water by osmosis and is fermented by bacteria, causing diarrhea. This is if the bacteria can use the lactose from the colon. Next, they say milk is a nutritious substance that provides several food groups. In areas of the world where milk is a main part of the diet, fewer people are lactose intolerant. Explain how natural selection has resulted in fewer people being lactose intolerant in areas of the world where milk is a main part of the diet throughout their lives. This is because they have varying alleles, and those people who cannot digest lactose, when they are children, they will not get sufficient nutrition, so maybe they die. Those who can digest the lactose are going to survive because they get nourishment, and so they will reach reproductive age and have offsprings. So I say the mutation occurred that led to variation in alleles. People with lactose intolerance were able to drink and digest milk, and this gave them better nutrition, and they survived to adulthood and reproduced, passing their alleles to their offsprings, and over generation, more have this advantageous allele. In part B, they say milk can be used to produce yogurt. The diagram shows a fermenter that can be used to produce yogurt in industry. So we can see there is a cooling jacket. Water comes in to cool and maintain the temperature. There is a stirrer for even distribution of heat of the nutrients as well as the bacteria, and of course agitating the milk. And here they say explain the role of the stirrer in the fermenter. Like I've already said, it mixes the milk evenly to ensure even distribution of nutrients and bacteria. It also helps to distribute heat and the pH to be even throughout the fermenter. Then they say explain the role of the water jacket in the fermenter. This is to cool so that the temperature can be maintained to optimum conditions. So I say the water jacket regulates the temperature inside the fermenter. Fermentation produces heat, and the water jacket removes excess heat to keep the temperature optimal for enzyme activity. Next, they say transgenic varieties of tomato plants have been produced that can photosynthesize more efficiently than natural varieties. To make the transgenic tomato plants, a gene is inserted into a vector that is then placed into tomato plant cells. Describe how a recombinant vector containing a gene can be produced. So here the vector I'm using is going to be a plasmid, and so we will get the DNA and cut out the gene of interest using restriction enzymes, and also get a plasmid and cut out using the same restriction enzyme, and then we will use a DNA ligase to connect or to combine the two pieces that are complementary. And again, using the same restriction enzyme ensures that when a cut is made, complementary sticky ends are left on either side. So I say the gene of interest is first identified and isolated using restriction enzymes that cut DNA at specific sequences. The same restriction enzyme is used to cut open a plasmid vector to create sticky ends that are complementary to the gene fragment. The gene is inserted into the plasmid using DNA ligase, which joins the DNA fragment, forming a recombinant vector ready to transfer into the tomato plant cells. In part B, the graph shows the effect of temperature on the rate of photosynthesis of tomato plants at two different light intensities. So as temperature increases, there is an increase in the rate of photosynthesis at both light intensities. However, here it levels off faster because there is low light, so light intensity is going to be limiting the rate. At higher light intensity, we see the rate increases with temperature. Again, it levels off. That could be because another factor is limiting the rate, because it's not going to be temperature, it's not going to be light intensity, then it could be carbon dioxide concentration. So here they say explain the effect of temperature on the rate of photosynthesis of the tomato plants at high and low light intensity. So I say that low light intensity, light is the limiting factor, so increasing the temperature has little effect on the rate of photosynthesis. At high light intensity, carbon dioxide concentration becomes the limiting factor. As temperature increases, there is more kinetic energy, so more frequent collisions with the enzymes per time, leading to a higher rate of photosynthesis. However, beyond the optimum temperature, the enzymes will denature because as we increase the temperature, and the rate is going to decrease or it will plateau. In part C, they say the diagram shows a sustainable glass house system that is used in the United Kingdom to grow tomato plants throughout the year. The glass house has artificial lighting and heating powered by electricity from a wood burning power station. So this means the wood can be planted and then burnt, so it can be kind of carbon neutral. Then they say carbon dioxide is pumped from the power station into the glass house. So the CO2 coming out of here is going to be used in photosynthesis. And then the electricity is also going to be used for lighting and for maintaining the temperature. They say discuss the advantages of growing tomato plants in this glass house system. In your answer, include the benefits for farmers and for the environment. So I say the glass house allows the tomato plants to grow year-round regardless of external conditions or climate. This could be external temperature, increasing crop yield and profitability for farmers. Artificial lighting and heating provide optimum conditions for photosynthesis, supporting faster growth and improved fruit quality. This is going to be for lighting, light intensity, and for heating, of course, it's going to be the optimum temperature. Carbon dioxide pumped into the glass house enhances photosynthesis, further improving the plant productivity. Electricity is supplied from a wood-burning power station, a renewable energy source which reduces reliance on fossil fuels and lowers carbon footprint. Waste products like carbon dioxide and water from the power station are recycled, making the system more sustainable and environmentally friendly. In question 10, the photograph shows a scarecrow in a crop field. We can see the scarecrow. They say scarecrows are models of humans placed into fields to stop birds from eating crops. Design an investigation to find out if changing the color of the clothes the scarecrow is wearing alters how effective the scarecrow is at stopping birds eating crops. They say your answer should include experimental details and be written in full sentences. Here we're going to state the conditions for the independent variable, the conditions for the organisms, these are the biotic factors. Then we repeat, then the measurement for the independent variable, time for the experiment, and certain standards that have to be controlled. So I said place identical scarecrows in different crop fields. The scarecrows must be wearing a different colored outfit. It could be red, green, yellow, and blue. Repeat in other areas and calculate the mean values, then count the number of birds that come to eat, or you could count the plants that have been eaten in a certain period of time. I chose 3 hours. The experiment should be at the same time of year, same wind speed, same temperature, and the crop study should be the same, and the field should have similar soil conditions and crop quality. So this brings us to the end of question 10 as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.